Right. Good afternoon, everyone. Today my talk is about the nanoneedles for influencing cellular behavior at the nanobio interface. So basically, what we refer here as vertical aligned nanoneedles, it is basically a nano scale protrusion from a bulk material, which can be a conical nanoneedle shape on the left or the nano tube on the right here. So basically, they have many applications in life sciences. So they can be used in biosensing, for advanced cellular studies, mechanical transduction, or our focus here, intracellular delivery. So one of the potential applications here is transfection. So CAR T cell therapy is one of the most innovative recent cancer treatment that re-engineered the patient's T cell for targeting and kill cancer cells. However, it can be costly due to the needs of using viral vector in delivering bio cargo into the cell. So nanoneedles are able to mediate delivery of various biocargo in vivo and in vitro. So it also has been shown to be able to deliver in diverse immune cells. So it makes it great candidates for replacing the viral vector used in the CAR T cell therapy. So before we go into the details, so how can we use these nanoneedles to influence cellular behavior and enhancing the delivery or the transfection? So the key lies in the cell nanoneedle interface, where we can basically control the geometry or the architecture of these nanoneedles, and we can influence the cellular behavior, such as increasing the viability, the proliferation, and also the transfection efficiency. So our group has successfully fabricated silicon nanoneedles with specific geometries. They are able to facilitate the gene delivery into various cell types, such as the GP86 cells, L1.2 Jurkat, primary mouse T cells. And also, at the same time, we fabricated the silicon nanotube that can also be loaded with various biocargo in the inner tube that can assist with the mediate the intracellular delivery and also enable the gene editing. This silicon nanoneedle platform shows a great potential. However, the fabrication process can be complex and it can be costly sometimes as well. So we're looking at an alternative for this. So we're looking at the polymer material, which, for example, the polystyrene, which has been used, like, for example, the Petri dish, and is basically used widely in most of the lab. It is easily accessible and is cost efficient. So if we can integrate the nanoneedles into this polystyrene material, and then we can create this cell culture tool for intracellular delivery. So our focus here is, this is the overall of the polymeric nanoneedles for bioactive cargo delivery. So first we have our nanoneedle platform, which also can be the needle or the nanotubes. And then we can load our bioactive cargo on the platform. And then we interface our cells on the nanoneedle. And then we can create this tight cell nanoneedle interface where achieving the intracellular delivery. So the first step of achieving this is first to create a silicon nanoneedle master as a template. So this template enables us to replicate it into various different type of polymeric material. So the first step is to deposit a polystyrene nanosphere on top of our silicon wafer using convective assembly to create this hexagonal closed pack array. And then we can shrink our polystyrene bits using oxygen plasma. And then this will act as a mask for our next deep reactive ion etching process. So after that, we can remove our polystyrene nanosphere and then do sharpening. And finally, we'll be able to obtain this silicon nanoneedle master. So we take this template and then we make it into a negative mold by using PDMS casting. So this mold can be used to replicate several different types of polymeric nanoneedles, such as SU8. Polystyrene, PDMS, with different kind of replicating techniques such as nano-imprint lithography, hot embossing, or just simply casting. So So one of the key questions here is if you want to understand how the stiffness affects the delivery efficacy, it's important to decouple the geometry and the stiffness of the nanoneedles. So here with this system, we are able to produce, fabricate 3 different types of the nanoneedles with the same geometry, but different stiffnesses, which is measured by the nano indentation here on the right video. And then after that, we can interface our nanoneedles with 2 different cell types, GP86 and L1.2, where they shown different spreading or retaining its globular shape on the nanoneedles. So we take these 3 different nanoneedles, 3 different types of polymer nanoneedles, and then we now do a messenger RNA delivery using this platform. So this messenger RNA is having a sci-fi tag, which can be shown in the yellow signal here, which showed a loading of this messenger RNA on top of our nanoneedles. So after interfacing with the cell, we can see the cell is uptaking the messenger RNA, and hence the cell itself showing the yellow signal in it as well. So because the messenger RNA is also encoded with the green fluorescence protein, so when successfully transfected by using this platform, the cell also will show green signal, which is here as well. So by using the flow cytometry, we are able to determine and quantify the transfection efficiency of using this platform. So we can see here with these 3 different types of polymer nanoneedles, we are able to achieve transfection. But interestingly, with different stiffnesses of these different polymer nanoneedles, it doesn't really affect our transfection efficiency much in the system. Also, we also show that this system are able to deliver and transfect the L1.2 cell as well, but with a lower transfection efficiency. So next, we use 3 different complementary techniques in the facilities to help looking at the nanoneedle cell interface to see what is the potential delivery mechanism involved in this delivery. So the first method we use is focused ion beam, SEM. So basically, we do a milling of the sample, and then we can see the cross-section of this nanoneedle cell interface. And we can see the deformation caused by the nanoneedles. And then also a closer look, we can see the ruffling of the cell membrane, plasma membrane. And this can potentially indicate that this nanoneedle will influence the intracellular signaling and then enhancing the messenger RNA delivery. Also, the second technique we use is to use ultramicrotome to slice the sample into a thin section for high resolution TEM imaging. So here we have the whole cells showing with the black dots, which is the nanoneedles. With a closer look, we can see the endocytic pits and vesicle forming around the nanoneedles. So combining with the immunofluorescence imaging at the bottom here, that's showing the clathrin heavy chain and caveolin 1, which is the endocytosis marker, and aggregating at the nanoneedle site as well. So all these show that the delivery mechanism can potentially be the endocytosis mediated delivery. So as a conclusion, we fabricated a highly tunable, biocompatible silicon and polymeric nanoneedles as a delivery platform. So, and then this platform will able to influence the cellular behavior and deform the cellular, causing some cellular deformation, and also enhancing the transfection efficiency as well. So ultimately, this platform are able to enable the nanoneedles integration onto the cell culture petri dish or cell culture ware for transfection application in the future. Yeah, thank you very much.
Fantastic work. Yes. obviously very proud of the progress you have made with this project. And we have quite a few questions that have come through on Slido. The first one is from Jane. How do you penetrate the cells? Is it by gravity of the cells only? And what is the viability of the cells after the delivery?
So, so with the transfection process, we actually use a centrifuge to pull the cell towards the platform. However, it's not necessary that the cell actually being penetrated by the nanoneedles. And then, yeah, basically it doesn't always show that the plasma membrane is like penetrated, but with the delivery of endocytosis pathways, it can enable the delivery of the messenger RNA. So with the viability, we tested the viability and it shows good viability of I think around 95% of the viability. So the platform is quite biocompatible, so it's not really damaging the cell.
Thanks, HaoZhe. There's a couple of questions from Ravi. How did the silicon pillars? How were the silicon pillars sharpened? What are the process steps involved?
Yeah. So in the nanoneedles can be further sharpened also by using the same deep reactive ion etching process by using the pseudo-Bosch process with a mixed gas. So that can be further sharpening the tip of the nanoneedles if it's not sharp enough.
What is the needle edge diameter on the silicon, and how is that diameter changed after transferring onto polystyrene needles?
Yeah, so the tip diameter is usually less than 100 nanometers in our silicon nanoneedles. So with the replication, you will lose slightly of the tip diameter into 250 nanometers. So 100 after replication, you lose about 150, but you can always sharpen it if required to get the exact same kind of sharpness.
Yeah, thanks. Because you answered very quickly, I'll just allow this last question from Jeff. How is the size distribution of the polystyrene nanospheres? How does that affect the needles? And how can you control that you have a single layer coating on the silicon wafer?
Yeah, so there is a bit of distribution of the size in the polystyrene bits because it's not a huge difference. This is commercially available polystyrene bits, and then we achieve this monolayer by using the convective assembly, where it's basically spreading the polystyrene bits into a thin film single layer on top of the substrate. Yeah.
Great. Thank you so much.
Thank you very much.
Please join me in